Hi, my name is Liam Darrow, and today I'll be reviewing the different organelles found in cells and their functions. The plasma membrane, or cell membrane, is a structure designed to separate the interior environment of a cell from the outside world. The cell membrane is selectively permeable. This means that, to an extent, it controls what enters and exits the cell. It utilizes two modes of transportation, passive and active transport. In passive transport, very small molecules are able to diffuse through the cell membrane without using any energy. An example of such a molecule would be carbon dioxide or oxygen. In active transport, larger molecules are moved across the cell membrane. In these circumstances, energy is required to complete the move. The other functions of the cell membrane are endo and exocytosis. These processes help to add nutrients or remove waste. In exocytosis, a piece of the cell membrane envelops a nutrient and is then fragmented off of the rest of the cell membrane in order to hold the nutrient. In an opposite manner, endocytosis removes waste from the cell with the help of the Golgi apparatus. Cytosol is a liquid found inside cells, and it's a pretty important goo. Obviously, there's very little structure to the cytosol of a cell. In fact, approximately 70% of cytosol is water. While cytosol itself has no primary function, it's the site for many other cell processes, such as the transportation of molecules. Ribosomes are a complex biological structure formed by a chain of RNA strands. They provide the genetic instructions for protein synthesis, making them an important part of any cell. All ribosomes have similar core structures in which the RNA chains form a base from which proteins form. Ribosomes are an important component of RNA translation. First, messenger RNA provides a series of codons to the ribosome that tell the ribosome what amino acids are needed, hence their prefix, messenger. The ribosome follows the series of codons, pairing the appropriate amino acid with each set using transfer RNA, or tRNA. tRNA carries anticodons that parallel the codons of the messenger RNA. The ribosome receives instructions from the nucleus, which has its directions transported by the endoplasmic reticulum. The nucleus may be one of the most important parts of a cell. Why? The nucleus contains all genetic material for a cell. Of course, this organelle is only found in eukaryotic cells. Prokaryotic cells do not have a nucleus. The nucleus is the largest organelle of a cell. It has an inner layer where DNA is kept and an outer layer composed of the endoplasmic reticulum and ribosomes. The nucleus provides a site for genetic transcription that is segregated from the location of translation in the cytoplasm, allowing levels of gene regulation that are just not available to prokaryotes. Gene regulation is a process by which the nucleus can control the production of a specific gene product. Within the nucleus is the nucleolus, which encircles specific chromosomal regions. The main function of the nucleolus is to transcribe and modify rRNA. The rough endoplasmic reticulum is associated with protein production and protein folding. The rough endoplasmic reticulum is rough because it is dotted with ribosomes that assist in protein production. This gives the sealed membrane a rough look along the edge of the membrane. The smooth endoplasmic reticulum is more tubular than the rough endoplasmic reticulum and forms a separate, sealed, interconnecting network. It is found fairly evenly distributed throughout the cytoplasm, and it is almost exclusively devoted to manufacturing lipids and occasionally metabolizing them. The Golgi apparatus packages proteins inside the cell before they are sent to their destination. This is especially important during endocytosis as the Golgi helps remove waste from the cell. The Golgi is made up of stacks of membrane-bound structures called cisternae. Enzymes within the cisternae are able to modify the proteins by adding phosphates and carbohydrates. These enzymes are unique to the Golgi apparatus and are an important part of protein synthesis. Mitochondria are the power plant of the cell. Mitochondria generate ATP, or adenine triphosphate, which is used in chemical energy transfers. Mitochondria appear as organelles within the cell and have their own DNA. This suggests that mitochondria were originally smaller prokaryotic cells that developed a parasitic relationship that eventually evolved into a mutually beneficial relationship with larger eukaryotic cells. The main function of mitochondria is to provide energy through the Krebs cycle and regulate the cell's metabolism. A major function of the peroxisome is the breakdown of very long-chain fatty acids. In animal cells, the very long fatty acids are converted to medium-chain fatty acids, which are subsequently shuttled to mitochondria, where they are eventually broken down to carbon dioxide and water. They have a simple structure, a lipid bilayer membrane, and occasionally a crystalloid core. Within the peroxisome are enzymes that assist in the breakdown of fatty acids. Human peroxisomes lack an enzyme that can break down uric acid. This is why uric acid within the body doesn't break down very easily. The cytoskeleton is a network of interweaved proteins found in the cytoplasm of a cell. Although the suffix skeleton implies a rigid structure, the cytoskeleton is not rigid. It is in fact a constantly changing structure as parts are added or removed. There are many functions that the cytoskeleton can perform. They provide support to the body of the cell and prevent deformation, and it's also involved in endocytosis and exocytosis as well. And it even segregates chromosomes during cell division. The lysosome is a spherical structure that contains hydrolytic enzymes capable of breaking down almost any biomolecules. These enzymes are originally created in the Golgi apparatus. They're released into the cytoplasm where they bind with endosomes, creating a lysosome. Once created, they begin to function as waste disposal by removing unwanted objects found in the cytoplasm. A centriole is a cylindrical cell structure that is found in most eukaryotic cells. They are used in cell division, specifically cytokinesis. They determine the placement of the nucleus in the cell and are a very important part of cell division. They also determine the placement of cilia and flagella in cells that have these organelles. Flagella are organelles found in both prokaryotic and eukaryotic cells used in locomotion and as sensory organelles. Prokaryotic and eukaryotic flagella have different protein compositions and designs, but they generally achieve the same goal, locomotion. Eukaryotic flagella are structurally identical to another important organelle, cilia. Cilia are divided into two categories, motile cilia and immotile cilia. Immotile cilia serve as sensory organs, while motile cilia generally create motion by beating in sync. The vacuole of a cell varies in its structures based on the needs of a cell, and the number of vacuoles present in a cell is also based on the needs of said cell. The primary function of a vacuole is to act as a sort of storage unit. They're like a garage. They all serve the same purpose, they just come in many different shapes and sizes. Finally, the chloroplast is a unique organelle found in plant cells. Their main role is to conduct photosynthesis, where the photosynthetic pigment chlorophyll captures the energy from sunlight and stores it in the energy storage molecules adenine triphosphate and NADPH, while freeing oxygen from water. They're structured so that the chlorophyll absorbs the maximum amount of sunlight and are highly specialized organelles. That's all, and thank you very much for listening to my presentation.